Hey everybody. So in my last recording, we went through messenger RNA and translation, the second part of protein synthesis, and we learned that the messenger RNA, when it leaves the nucleus, is going to go over to the ribosomes in the cytoplasm. So it'll find a ribosome, and that ribosome might be on the rough ER, or it could be free floating in the cytoplasm. But it's going to go up there and get read 3 at a time. So the ribosome and the the ribosome and the ribosomal RNA will work together to get this read 3 at a time. And what happens is a third kind of RNA, which is the transport RNA, will transport an anticodon, which is the complementary code. I can't hold all this. Yes, I can. The complementary code. And so as these get read 3 at a time, 3 at a time, 3 at a time, what's going to happen is these start lining up. Well, the anticodon is bringing an amino acid. So this is how stuck with tape. I'm going to grab some arbitrary ones that aren't tape. So these just start lining up. And so every amino acid has this same frame, and the kind of the arrow looking part here will connect to this triangle looking part, and these link up into a very long chain. And that chain creates the primary structure of the protein. So the primary structure is just the straight line. So if each one of these, or the sequence, the straight line, if each one of these push pins represent one amino acid, so it's going to represent this, not this. So we're moving away from this idea of the code for right now. So if they say that first yellow one represents the methionine, then and each one of these represents another amino acid. You got 20 possible amino acids in your body. This lineup, just this straight line, the sequence, is referred to as the primary structure of the protein. And then the next thing is a secondary structure, and that has to do with what attracts to what within this strip. So let's say that, let's make a rule saying that these green push pins represent an amino acid that likes to bind together. Particularly if there's 2 in a row, they're gonna really hug up close to each other. And so I just happen to have that in a couple places here. So I'm going to bin that. And then let's say if I have 3 of these in a row, that's going to cause a bend. But in this particular bend, they're going to arrange in a way that what's represented as these representing amino acids as these push pins, they're going to turn the other way. And so that, and then we'll make another rule. We'll say whenever 2 greens and 2 purples are together, that causes it to kink kind of sideways. And maybe in a way that it kind of turns upward. And so what we have here is, first when we start getting the bending, I moved ahead a little. So if we just have it all on a single plane, we're now into secondary structure of the protein. And then if we start moving it into 3 dimensions, say like this, then we'd say that is tertiary structure. So we've almost got a fully structured protein, I guess you could say, except there's going to be another possibility, and what that's going to be is a quaternary structure of the protein. So this, by the way, looks a fair amount like an antibody, okay? Protein made by white blood cells. So if this were an antibody made by a B cell, then what this, the function of this thing would be to tag onto an invader. So this, invader. I had an invader here, but I think the dog ran off with it. I was going to use her toy. So let's say my hand's the invader, and the purpose of this type of antibody is just to tag onto that to say, to signal other proteins to come and cluster around and start disassembling this, and also to bring in more white blood cells of different kinds, and some of these white blood cells will start also disassembling. Okay, so but this isn't quite ready, because if this were it's kind of like a slingshot, say, but it's not quite ready because it easily could fall apart, bend apart. So it must need more structure to it. So that's where we get into quaternary structure. I'm going to have to reach. Oh no, dropped on the ground. So quaternary structure is when you have, this one doesn't have push pins, I didn't put that together because they get kind of pokey, but just imagine that it too is made up of a sequence of amino acids, and this is going to represent another protein that is part of this antibody that is part of the structure. And now we have a quaternary structure. So quaternary structure is, wrap as nice as I wanted it to, but you know, here, let me fuss with it for just a minute. Smack myself in the face with it. All right. And so now we could say that's the quaternary structure. So quaternary structure is when multiple proteins come together to form the full product of what's being made. And that will generally, that occurs at the Golgi apparatus. Remember the Golgi apparatus takes protein products and packages them. Well, here's a package. It's actually a protein that's been completed by having different proteins added on. It might have 3 or 4. So a protein that's of interest in current events these days is the protein on the outside of the COVID virus. And I'm going to post a picture of that. The outside of the COVID virus has what we call spikes, and the spikes serve as keys to get into the cell. And we've talked about viruses and their keys. I'm just using that as an analogy. So they are referred to as spikes. Well, the spikes are protein. So you can see in the picture the quaternary structure of these proteins, because the different proteins are being shown in different colors. So there's work being done when we're trying to figure, when scientists are trying to figure out vaccines, how are we going to slow down this virus? A lot of what they're looking at is how can we keep these spikes from docking on a receptor on your cells and entering. And the receptors that they dock on are receptors that are found along the epithelial or lining of your lungs, particularly deep in the lungs. And they've also found that there are some of these types of receptors within the nasal cavity too. So that explains how COVID can get into the body quite quickly. And so, yeah, there's a lot of work on, well, what can we do? So let's just say, I'm going to pause for a minute. I had to get to what I wanted to use. So let's say this is the COVID virus, and that these are the spikes or the keys. And so some different things that researchers are looking at is, can they create a receptor-looking kind of attachment that will tie up these keys so that they are so busy just latching onto things that won't give them access to your cells, that they don't get into the cells, or certainly don't get in as many numbers and don't get in as quickly. That slowing it down could help your immune system then recognize and fight them off. And another thing that we're looking at, of course, is vaccines that could maybe increase the immune response of another type of antibody, which is the T cell. And a T cell is more aggressive than a B cell antibody, because a T cell, Thunder's not doing this on cue. Get it? Now she doesn't want to get it. We're playing a little tug of war. So a T cell attaches, but it also tries to disrupt the spikes. So where a B cell antibody would latch on to an invader, and that would help alert other blood cells to come and create a strong force of resistance. Get it? Earlier she wouldn't stay off of it, now she doesn't want it. The T cells are a little more aggressive and can actually disrupt the movement and remove, smack myself in the face, viruses, or disable them so that they cannot do their damage. Now, the preferable mode of getting rid of this virus or slowing this virus down is to attack these spikes before the virus ever enters your cells. So it'd be in your body, but between cells and not getting into the cells. But they are also, as I mentioned in the last talk, they are also looking at mechanisms for dismantling the production of viruses once they're inside your cells. You can imagine if we can just get them before they ever get into your cells, then that would cause less damage to the body, prevent viruses from making more viruses and spreading. So this is the ideal spot of where we want to push research forward, is in trying to keep the spikes from being able to enter through endocytosis after they attach to a receptor and then allow themselves into your cells. And she's off. And so in the next part, I'm going to talk about moving away from coronavirus, the COVID-19 virus. I want to talk about going back to the code, going back to DNA code, RNA code. What happens if the code changes? So okay, so thinking about what it is that causes proteins to be able to act the way they act and do their jobs, it hopefully is becoming apparent that the sequence really has everything to do with the form that the protein takes and therefore the job that it can perform. So let's say I'm missing, instead of one of these greenies, and the double greens are causing a bending here. But let's say instead of this one, I've got some other color, there's an orange there. I mean, what if something's flipped? And so maybe in this scenario, it doesn't hold, it holds together a little, but not quite so tight, and that could very much affect how this molecule functions. So the code allows for the lineup that allows for the secondary, tertiary, quaternary structures to form based on attraction of certain molecules together or repelling of them, you know, whether they're polar, non-polar, so if they have a positive, negative charge, those sorts of things. And so what happens when the code changes? Well, first, code changes every generation of cell. Every time a cell replicates its DNA to make a new cell, there's going to be some error. So we have an underlying error rate that is predictable for cells, and at times there can be reasons like radiation. There can be reasons that would cause that rate to go up. So certain circumstances could cause error rate to go up, but there's always going to be a certain amount of mutation. And that's both true in the body cells, the DNA in the body cells, and also the DNA in our gametes or our reproductive cells. So some of mutation will just be within our own bodies over time as DNA is copied so that cells can make more cells. But then perhaps more importantly for the bigger picture is that mutations also occur within the gametes or the sex cells. And that's a good thing, because that's where we get all the variation in the world of living things. And so I wanted to talk about, just so you can kind of see how a little change could cause maybe not a big deal or a huge deal. So if we just go back to thinking about the alphabet, I'm going to, instead of using DNA or RNA alphabet, I'm going to use an alphabet we're more familiar with, just to make a quick point. And I'm going to write a quick sentence. So if we just imagine that this was DNA code, and this was just a sentence in a book, very simple book. Then if I were to make the kind of a genetic error that we would call a replacement, maybe the fat cat becomes the fat rat. It changes some meaning, but if you thought about it in context of a book, probably through reading other sentences, putting it together, you'd figure it out. Oh, thunder. That was fun. And that was kind of hilarious because I don't know if it showed up on screen or not, but the dog just chased my slightly heavy cat across the yard a little bit. So in context to the storyline, you'd be able to figure it out. Now, what if, so that's a replacement. That is a replacement mutation. And then there's another kind of mutation that we would call a frame shift. So a frame shift is when there is a deletion or an insertion of a letter. So I'm going to write that again. And because, if you will please remember that codons read in sets of 3. So if this were my DNA code, like if this was TTA, CGC, TAC, you know, if this was DNA code, then we remember that it is read in sets of 3, and when you did your marking up of the genetic code, you would draw on your lines through like this. It's really hard for me to do backwards. Okay, but what if we add a letter in? So if you had any struggle at all with that last assignment, that probably worked in your advantage, because the struggle would mean that you probably have already experienced this a little. If I were to add a letter in, and I will just put it in at the beginning for yucks and grins, I could put it anywhere. And now we know that I've got to change this. So now my sentence becomes, this, doesn't it? Okay, and then I'd have to draw the lines between, and we have something nonsensical. And so that could be an entire gene from the addition or deletion, because you can try this with a deletion too. All right, if I were to take the T off, then we'd have to be marking differently, and we end up with something nonsensical. So a frame shift type of mutation can mess up an entire gene. Now that would be reset on the DNA strand by the time you come to the next gene, and the non-coding, because there's non-coding DNA in between the genes, it wouldn't affect the next gene, but it could affect an entire gene. Whereas a replacement might change. It might change a little bit, but it could be a bad deal. But oftentimes a small change like this, just the body works that out. And in fact, if you go back to your codon chart and you take a look at that, you probably notice that there are multiple codes that code for the same amino acid. So I'm going to show you that here. Let me pull that up. So if you recall, so let's see. So this is arginine, and so CGU, CGC, CGA, CGG, all of those code for the same amino acid. So you find along the wheel that there are several ways of getting the same amino acid, and that can be that is a way that the body actually allows for there to be a little bit of coding error without there being disasters. And then this frame shift, and of those two, frame shift is most likely to cause a bigger deal. The smaller mutations may have no effect. They could have a positive effect. They could have a negative effect. But oftentimes they have a silent effect. We don't even notice that they're there. And back to the example of the amino acid puppers. So let's say we have one that disassembles a little more readily. So it could be that holding very tightly, that it will be able to keep its construction even in, say, warmer temperatures, because like when we have a fever, it might hold together better than this one that might be looser. Or it could be that for some reason, when you have a fever, releasing this and changing the response of the antibody, maybe that ends up being a good thing. Maybe that causes, hey, come here. Maybe that the B cell antibodies are going slower, for all we know, could cause the cell antibodies, the dogs being the T cell antibody, to come up in fuller force and take over and save the universe, yeah, and help you get over a infection, theoretically. So there are a lot of things in motion when we're talking about genetics. So we'll be digging into genetics starting very soon. We're going to talk a little bit about how cells do divide and how it is we get variation of genetics when, from our moms and dads, how we get scrambled combinations of those and brain freeze. For this section, you have a short quiz, and then also there is a worksheet for you to read through and answer a few questions regarding mutation. And so when our next subject will be released this week, and that's going to have to do with cellular division and how it is that genes get replicated and moved into different sides of a cell, so that when the cell splits, the genetic content is ready to be divided in equal amounts. So, and we'll be comparing what that looks like in body cells versus what that looks like in reproductive cells. All right, so have a great day, everybody, and I'll talk to you soon. Oh, and don't forget Zoom meetings on Tuesdays, Tuesday afternoon at 2 o'clock. All right, see you later.